Hello students, very good afternoon. So before we start today's session, where we are going to start a new chapter, so can you just give me an assurance or tell me if my audio and video is proper? And you can please use the chat box. Please mention in the chat box if my audio and video is clear for you so that we can continue the session. Okay, students, are we cool to go? I think we'll start the class today. Yeah, thank you for texting. Somebody texted. Let me see. Yes. Yes, thank you very much for your reply. It's very helpful. So let's start our session. So before we start today's topic, it's very, very interesting because we are talking about body fluids and circulation, where many of the people get attracted to this part of human physiology because it's about heart. And whenever we talk about human physiology, somewhere we are drawn towards the structure of heart and the functioning of heart, apart from even more complex organisms like neural control and coordination, right? So anyways, so first we will start with the outline of this chapter. So in this session, we'll be talking about few important components, where first we'll talk about what are body fluids. And in body fluids, we'll talk about what is blood and its components. So in blood and its components, see, blood is a mode of transportation of substances across our body, and it is a vehicle. So we have to study the components of blood and how the blood helps in being a part of the circulatory system. Okay, so that will be that. Today we'll be only talking about blood and its components, and moreover, in blood and blood components here, that is blood grouping and coagulation would be another important topic we'll be covering. So after knowing this, we're going to conclude this session, and in the next session we will talk about the human circulatory system. All right, students, it's been a custom, so we'll start with a question before we start the session. So you have your first question. In blood, WBCs are more than RBCs, RBCs are more than WBCs, RBCs are less than platelets, platelets are less than WBCs. So which of the following is correct when it comes to blood? So you have your question. So you can use your chat box to answer the question. Okay. So your time starts now. Okay. So we have one answer. Come on. The rest of the students, please answer. Students from YouTube, you can use your chat box. Yes, we have more answers coming up. That's very nice to know. Okay. All right, students. So we'll go with the answer. So in blood, see, in blood, we have two components. One is the plasma component, other one is the formed element components. So if you see the formed element components, it is made up of cellular parts of the blood, which includes red blood cells, white blood cells, and you have platelets. And red blood cells are very small cells, and they're very, very important to our body because they act as the vehicles for supplying oxygen as well as removing carbon dioxide. Okay. So if you see the cell count, red blood cells are the highest number of cells that is present in our blood. Next it comes is platelets, and then followed by white blood cells. Okay. So our answer for this question is red blood cells are more than white blood cells. That is option B is the correct option. Okay. So anyways, you have your second question now. RH factor is named after monkey, Drosophila, rat, and human. Like man. So choose your correct answer, and you can again use your chat box to answer this question. Yes. We need more answers. It's a very simple question, very simple and straight question. If you don't know, or if you don't know also, you try to attempt it, because as you attempt, you build a stronger memory when you know your mistakes. Okay, it's nice. So, all right, we have so many important topics to talk about. Let's get into the solution part of it. RH factor is rhesus factor. Okay, and it was first identified from a monkey called as rhesus monkey. Okay, so that is why RH factor is named after a monkey called as rhesus monkey. Okay, so we will talk about what is RH factor and how it influences the physiology of humans, especially during birthing stage, okay, or during pregnancy. So our answer for this question is option A. Okay, students. So let's get back to our session. That is, we have to talk about body fluids, first of all, and then we'll talk about circulation. So if you see all living organisms, do they use any of the mechanisms or modes to transport substances across their body? And the answer for this question is definitely yes. Every single organisms on this planet, they use some of the other modes to transport substances. Now, when you talk about unicellular organisms, it is quite a different case in them, because it is a simple diffusion across the body surface can actually accomplish a lot of purposes. But when it comes to multicellular organisms, from cellular level organism all the way till the complex organ system level of organization, like you have sponges, coelenterate, and you have your platyhelminthes, okay, aschelminthes, and then you have arthropods, and then you have higher organisms like aves, reptiles, and mammals. You take any of these organisms, they have some system in place where they use it to transport the substances across the body. Now, why do we need these medium for transportation? Let us take an example of our blood. So what does our blood do? Is our blood just pumped to all parts of the body just because it supplies oxygen and it carries carbon dioxide from the tissues and back to the lungs? It's only one part of the job of the blood. What happens when you eat food? So you eat food, right? So the food that you eat goes through your alimentary canal. It goes through your stomach, it goes through your small intestine and large intestine, and through basically the waste is eliminated through rectum. What happens during this process? Is there any important thing that is happening during this process? Definitely yes. If you see your small intestine and large intestine, there is enormous amount of absorption process taking place. So where does this absorbed nutrients go, and how does this absorbed nutrients get transported? Right? So this absorbed nutrients get transported through blood, isn't it? So our cells, they perform a lot of metabolic activities for their growth, for their functions, for their repair, for their cell division, and etc. While they do this, they require energy. And if they have a requirement of energy, definitely they will have to undergo metabolic process. And the resultant of this metabolic process is the production of waste, correct? No? So they produce waste, very toxic waste, for example, urea, uric acid, ammonia. So these waste are very bad for our body. It can be cytotoxic and it can kill our cells. So how do our body use this mechanism to collect all this waste from almost 30 trillion cells in our body? And how do they filter out through one system, correct? No? Our blood goes through our kidneys, and our kidneys filter our blood and removes all this toxic waste, and it will be eliminated in the form of urine. So you can see that the blood has more than one role to play than we think that it does. So that is why all the living organisms, which are like complex living organisms and simple living organisms who are multicellular, they have built this mechanism of circulation and the indulgence of these body fluids. Okay, so these body fluids are popularly blood. But what about other organisms which are in the lower category? Take, for example, sponges and coelentrates, which are highly lower category of organisms with cellular and tissue level of organizations. So what do they do? Do they have blood? Definitely they don't have blood. So then how do they circulate things across their body? They use water as a medium for circulation. If you see sponges, they have this canal system, where the water is pumped through these small pores like ostia, and it goes through osculum, which is, see, you can see this is the osculum, okay, the bigger pore. And the small dot-like structures are called as ostia. So the water enters through this and it exits through the bigger pore. And during this entry and exit, there is so many things that is happening between this small, gap. It can be fixing of the gaseous requirements. It can be exchange of nutrients, or it can be removal of waste, okay, etc. But when it comes to higher organisms, most of the higher organisms, okay, they use blood as a mode of body fluid, okay, for movement or transportation of substances. So on the next one, among the blood, we have one more part is called as lymph. So what is lymph? See, our blood, for example, it's a very beautiful image here. So we have our blood vessels, and we know that our blood vessels get thinner and thinner as it keeps on debranching. So it gets to a level of capillaries. It becomes so small that it is microstructures. So when it comes to the capillary level, our red blood cells will no more be able to move forward, or it will not be escape outside these vessels. Now, during this point, what happens? Our blood is tremendously made up of higher amount of content called as blood plasma. And the blood plasma leaks out of these blood capillaries and reaches our interstitial spaces of our tissues. Okay, it goes between our cells and tissues. Which one? Blood plasma. And whatever the plasma that is leaked out of our blood vessels, it has to return back to our main blood, correct? No? Otherwise, what will happen to this? Our blood will lose its composition. So if the blood has to come back to its original, it has to reunite, who will help in that? The lymphatic system, or the lymph, which helps in that, okay? So usually lymphatic system is very, very responsible for carrying gaseous substances as well as nutrients across our body. Okay, people. So let us see what is our blood made up of and what are the components of our blood. Okay. So when we take into consideration of blood, so let us assume that we conduct the process of centrifugation. So what will I do? I will take around 10 to 15 ml of blood and I will run centrifugation. If I run centrifugation, my centrifugal tube or my centrifugal tube will divide my blood into two important zones. So what is that? One will be a very thick red color zone, okay, very red, okay, very thick red color zone, and there will be a white buffy zone, okay, there will be buffy zone, small buffy zone, which is like cloudy buffy zone, and majority of it will be yellow straw color liquid called as blood plasma. Okay, we have plasma, which is yellow color. And then we have buffy coat. Okay. We have buffy coat. And then we have cells or formed element. Okay, people. So now let us see what is present in our plasma. If we consider the blood plasma is composed of cocktail of substances like amino acids, nitrogenous wastes, nutrients, electrolytes, okay, like ions. Then you have proteins and gases like oxygen and carbon dioxide. So these are the dissolved substances in your plasma. Whereas some of the important components of your plasma is the type of proteins it holds. So what are the type of different proteins that our blood plasma has? Albumins, okay, then globulins, and then fibrinogen. So these albumins, globulins, and fibrinogens are very, very important for the composition and what the blood is capable of doing. Okay. So this is very, very important. Albumins, fibrinogens, and then we have globulins as the type of proteins that is present in our blood plasma. Is it understood? So next one, when it comes to our formed elements, in the formed elements, basically we have two coats, isn't it? We have the buffy coat and then we have the cellular region. In the buffy coat, you have a lot of your WBCs or leukocytes. Okay. Whereas when your cellular zones, that is the formed elements, you have your erythrocytes and platelets. So what is your leukocytes? Leukocytes are your WBCs. In leukocytes, you can divide it into two important categories. What are those? One is granulocytes, other one is agranulocytes. So when we observe in microscope, okay, so the cells which are having a lot of granules in their cytoplasm, okay, if you take the cell, and within the cell cytoplasm, there are lots of granules that are present. So you can easily observe it under the microscope. These are called as granulocytes. So the number of granules are very, very high. Whereas when it comes to agranulocytes, they also possess some granules, but the number of granules are very, very less compared to that of granulocytes. Okay. So like grain-like structures. So in granulocytes, you have three important cells. One is neutrophils, basophils, and eosinophils. So neutrophils, they are neutral in pH, basophils are basic, eosinophils are acidic. So based on what type of stain they react to, for example, eosin is a stain that these cells react to. That is why we call it as eosinophils, and eosin stain is acidic in pH. Basophils is basic in pH because they react to the basic stains, whereas neutrophils react to the neutral stains, okay? And that is why they are called as neutrophils. Whereas when it comes to agranulocytes, there are two important types of cells. Those are called as lymphocytes and monocytes. Okay, students? So these are the formed elements. All right. So as I said, if I take the composition of blood, if I consider the composition of blood, and if I conduct the centrifugation process, so I can, 1 second. If I consider the composition of blood, I can conclude it into two important factors. One is the plasma part of the blood, other one is the formed element part of the blood. See, I told you, right? If I take a centrifugation of the blood, I will get three zones. What are those zones? One is the red color component. Next one is the buffy zone. And then I have your straw color, a straw color zone that is called as a plasma. So this yellow straw color region is called as plasma. And what is the composition of it? Out of 100% of the blood, 55% of the blood is made up of plasma. Okay. And the pH of the plasma is almost neutral, but little alkaline, that is around 7.4. Okay. So plasma acts like a fluid matrix. So plasma holds all the cells, all the formed elements, okay, whether it is WBCs or red blood cells, all of them in one place. Okay. Whereas when it comes to formed elements, around 45% of our blood is made up of formed elements. That includes your red blood cells, white blood cells, and then your platelets. Okay, fine. So as I said, if you take plasma, one of the major constituents of the blood plasma is water. Okay, majority, around 70% of the plasma is made up of water. And after the water, one of the major constituents of our blood plasma is the plasma proteins. Okay, I told you plasma proteins determine the characteristic feature of our blood. So that is, if you take fibrinogen, it is responsible for coagulation or clotting nature of our blood. If you take albumin, they maintain osmotic balance and regulate blood pressure. If you take globulin, they act as antibodies. Okay, very, very important features for a blood to have. Okay, next, when it comes to the rest of the parts that are dissolved in your blood plasma, that would be glucose, amino acids, lipids, cholesterol, and which help in providing the nutritional factor for your cells, and other factors like inorganic ions, which maintain the electrolytic balance in our body, okay, for regulation of osmosis, such as sodium, magnesium, bicarbonates, calcium, and chlorine are also present in our plasma. And dissolved gases like calcium, carbon dioxide, oxygen, and nitrogen are present, which play a role in transportation. Okay, when it comes to formed elements, as I told, red blood cells are very, very high in the overall ratio of formed elements. And next comes platelets. Okay, and then we have white blood cells. So in the white blood cells, as I told, we have granulocytes and agranulocytes. So let's see their characteristic features. If you talk about red blood cells are red because they have a metalloprotein called as hemoglobin. And the hemoglobin protein is responsible for carrying the oxygen molecules as well as the, fixing certain amount of carbon dioxide when it comes to transport of gases. Okay, so because of the heme protein, our red blood cells gain this color of red. And one of the characteristic feature of our red blood cell is our red blood cell is biconcave in shape. Okay, it is biconcave in shape. And the biconcave in shape is one characteristic feature which helps in binding of gases to the red blood cells as efficient as possible. Okay. And if you see the cell, red blood cell, our red blood cell is made up of a membrane similar to that. It's a plasma membrane, but the plasma membrane of the red blood cell is made up of a special type of protein called as spectrin. Okay, spectrin. And this protein, spectrin, is so helpful for the red blood cells. Why? Because when the red blood cell squeezes into smaller and smaller blood capillaries, it has the capacity to change, twist, turn, and fold into smaller components. And then when it is again pulled back into the bigger blood vessels, it will regain its stage, its form. Okay, it will regain back its shape and form. So that elastic nature of the red blood cells is because of this component called as spectrin. Okay, so if you see the red blood cells have a very short lifespan. They have a lifespan of only 120 days. So usually they are enucleated cells. The reason is because when they are formed, they have nucleus, and as they mature, they will lose this nucleus. Okay, and they mature. So after the completion of 120 days life cycle, they will go to the spleen, and then they will be degenerated, and the components of the dead red blood cells will be used up or recycled for the construction of new red blood cells, etc. Okay, so if you see the count of red blood cells, they are up to 5 to 5.5 million cells for every cubic centimeter. Okay, so where are they found? They are found in the red bone marrow tissue. So red bone marrow is found in your long bones, especially your the bones of your arms, the bones of your legs, the bones of your pelvis or the pelvic girdle. So that is where the red bone marrow is present, and that is where the red blood cells are formed. Okay. So if you see, as I told, if you see the structure of the red blood cells, they are biconcave in structure, and they do not have any cell organelles such as nucleus. Okay. And then they don't have mitochondria or Golgi complex. Okay. Their function is to carry carbon dioxide and oxygen transportation, and their graveyard where the red blood cells die or recycled is at the spleen. Okay, spleen is also called as graveyard of red blood cells. So let's come back to the white blood cells. We know that our body is very well protected by our immune system, and immune system is a mechanism of our body to be able to understand the difference between who is me and who is others. For example, if you are in your family, you know who are your family members and who are not your family members. Similarly, who are your classmates and who are not your classmates. Imagine a junior, or if your other classmate, other class guy enters your class. So you would be immediately able to recognize, okay, this guy is not from my class. How do you recognize this? So what does this help you in? It will help you in defending your territory. It will help you in, recognizing what is me and what is not, me. So these white blood cells are very, very important for that feature. So they provide your body with immunity by protecting it from any of the foreign agents. Okay. So if you see white blood cells, they're quite colorless in nature, and they have a short lifespan of 1 to 15 days. And the number of white blood cells are very, very important in determining the healthy state of a human being. More the white blood cells are there, that means the body is under some infection. Okay, some of the pathogens would have been present in your body. That is why your body is producing more of these cells to defend itself. So in a normal healthy state, the number of red blood cells is around 6,000 to 8,000. Okay, cells for every millimeter cube. Okay, so where are they formed? They are formed in bone marrow. Okay, they are found in basically one of the important cells. Okay, regions called as red bone marrow. That is your bone marrow as well as your spleen. Pelvic girdle. Apart from here, they go under a process of maturation, and they go the process of differentiation in several of these lymphoid organs such as spleen, lymph glands, etc. Okay, so bone marrow, lymph glands, and spleen are some of the very specific locations for the formation of white blood cells. So when it comes to the structure of white blood cells, the morphological structure of white blood cells is very, very important criteria in differentiating what type of cells they are. Okay, So one of the major category is the type of the nucleus they have. So they are nucleated and they come in different structures. Okay. So when it comes to function, as I said, they play a very important role in our immunity. Okay. You can see here, based on the type of WBCs, we can calculate, we can differentiate them into two types, right? One is granulocytes and other one is agranulocytes. If you take an example of neutrophil, right? Neutrophil is a granulocyte. So if you see neutrophil, can you see here? The nucleus of this neutrophil is lobed. The number of lobes can increase from 3 to 5. Okay, it can increase up to 3 to 5. So what does this neutrophil do? It helps in phagocytosis. What is phagocytosis? So this is a very generalized immune response. Whenever somebody comes, what do? I will capture them, I will neutralize them, I will digest them, and I will eat them. So that is called as phagocytosis. So what is that percentage of granulocytes? Up to 60 to 65% of the granulocytes is made up of neutrophils. Next comes eosinophils. So these eosinophils react to this eosin stain, which is acidic stain. So they fight against these parasitic infections. Okay. So usually when you have any of the parasites that have infected your small intestine or anything, so their count is very, very high. Okay. So what is that percentage? Only 2 to 3% of your eosinophils are granulocytes, and their nucleus is also lobed. And the number of lobes depend from 2 to 5. Okay. When it comes to basophils, basophil are present. You can see no more number of basophils when there is an allergic reaction. Okay. So allergy means whenever you are exposed to dust, whenever you are exposed to certain food, okay, you get rashes, itching, eyes will become very, very watery. Okay. Your nose burns. So all these are called as allergic reactions. So in these allergic reactions, your basophil will play a role in that. Okay. And they are up to 0.5 to 1% out of your total granulocytes. Understood? So when it comes to agranulocytes, you have two very, very important cells. One is lymphocyte, other one is monocytes. When it comes to lymphocytes, lymphocytes will produce further, okay, more types of cells such as B lymphocyte cells, T lymphocyte cells, and NKC cells are also called as natural killer cells. So what are B lymphocyte cells? B lymphocyte cells will produce proteins called as antibodies, which help in recognition of what is what. That means it helps in recognizes whether it is bacteria, what type of bacteria, what type of disease it causes, and how do I neutralize it, etc. When it comes to T lymphocytes will synthesize certain type of cells which help in defending your cells based on the messages that is given by B lymphocytes. And natural killer cells are very vague in their immunity. So they just attack like your phagocytes. They just go and recognize certain cells which are infected and they will neutralize them. When it comes to monocytes, they are specifically built for fighting bacteria, virus, and fungi, and they are up to 6 to 8% in the overall amount of agranulocytes. And as I said, students, the number of granules are very, very high. You can see here, no, you can see these granules are very, very high in granulocytes, whereas agranulocytes also have granules, but the number of granules are very, very less. Is it clear? Fine. One second. All right. So students from YouTube, I hope you're understanding. Okay, you can just put a message if you have any doubts. All right. Fine, people. So our next interesting topic for now, okay, is we have to talk about something called as coagulation or clotting of blood. So what is clotting of blood? So whenever we have an injury, our blood flows for a certain duration of time. And after this, slowly the bloods will stop running and we will have clotting. I mean, like the blood will stop flowing and it will seal itself. What if there is no stopping mechanism for the flow of blood? It would be so hard. Even if a mosquito bites you or if you have a small cut, the blood would keep on flowing out of your body and ultimately it would lead to your death. So that is why our body has an inbuilt mechanism for the clotting of blood. Okay. So when you talk about clotting of blood, we would have very it's very famous, right? Clotting of blood is very, very famous because we talk about how it helps in preventing the loss of blood during the state of injury. But if you see the overall clotting process, it is not just one step process, it is a multi-step process, and it is a very elaborate process. So whenever we talk about clotting of blood, immediately we jump into coagulation pathway, where we talk about some of the clotting factors and how it helps in clotting of blood. But when you see what is the stage wise mechanism our body deploys in really stopping the blood flow, okay, it happens in 4 different stages. Okay, so what is the 4 different stages? For the clotting to even begin, first what you have to do, you have to fall down or you have to have a cut. So that is called as injury. First, when you have an injury or whenever a blood vessel is severed, okay, now what will happen? There will be flow of your red blood cells, white blood cells, all of them will be leaking into the gaps. Okay, it will be coming out. Okay, it will be flowing. So as this happens, now what would you do? Imagine that you have a tap. Okay, you have a pipe and you're gardening or you're filling a pot or something like that. Now you want to immediately stop the water flow from that particular region. What will you do? You can't run like 100 meters back and close the tap because you don't have that much time. You need an immediate mechanism to stop the water flow. What will you do? You will squeeze the pipe, isn't it? You will squeeze the pipe. So how do you squeeze the pipe? You just hold your both the arms and you try to squeeze it so that the amount of loss of water is less as you're watering. So can our body use the same mechanism? Definitely yes. So once you have an injury, now what our body will do, it will deploy a mechanism called as vascular spasm. So what is vascular spasm? If you see our blood vessels is made up of 2 different type of muscles. One is circular muscles, other one is longitudinal muscles. Now what these muscles will do, these muscles, they will contract. Okay, your circular muscles will help in reducing the size of the blood vessels. See, you can see that this is the normal size of the blood vessels and this is the reduced size of the blood vessels. Okay, whereas your longitudinal muscles, what they will do, they will withdraw, or they will see, if this is your blood vessel, what it will do, it will pull them away from the injury site. So when that happens, this is called as vascular spasm. When the vascular spasm happens, when the contraction and withdrawal of your blood vessels happen, there is a reduction in blood loss. Correct? No? So we understood the first mechanism, that is vascular spasm in the clotting of blood. So second one is platelet plug formation. What is platelet plug formation? Now, if I have a blood vessel, now I cut here. Whenever I cut here, my blood vessel is made up of several type of proteins. So one of the important proteins that are present in our blood vessels, in my blood vessel or in all of our blood vessels, is our collagen. Okay, so there is collagen fibers. So whenever these collagen fibers are exposed because of the injury, now what will happen? This will trigger or this will activate the platelets. Usually in our blood, the platelets are very, very soft. Okay, they're very, very soft. So these soft platelets, when they are exposed to these collagen fibers in our blood vessels, what they will become, the soft platelets will become sticky and spiky. Okay, what will they become? They'll become sticky and spiky. When they become sticky, means gluey. When they become sticky and spiky, they will come and attach to each other. So as they come and attach to each other, more and more of your platelets keep on getting sticky and spiky, sticky and spiky. So if you are having one platelets and the rest of the platelets are also becoming sticky and spiky, now what will happen? They will start forming a huge ball of platelets, right? Like a plug, right? What will you do? If you are having a leak tap, you would go and plug it, right? So similarly, in your platelet plug formation, because of the platelets releasing some of the chemicals to attract other platelets, they will start forming a sticky and spiky platelet plug, and they will block this injury site or the blood vessels. See, once the platelet plug is formed, it is not the end of it. This is the beginning of it. Why? Because this is a very, very, very temporary a mechanism to stop the loss of the blood. Okay, so it cannot hold, it cannot repair perfectly the flow of the, the loss of the blood. Next process, the very elaborate and very detailed process where the loss of blood is prevented is by coagulation. Okay, the next process is coagulation. What is coagulation of blood? It is a cascade mechanism. Okay, why do we call it as a cascade mechanism? Because the process of coagulation includes, okay, fine. The process of coagulation includes an elaborate mechanism of several proteins. Okay, see, if you see the components which are participating in coagulation, there are around 13 factors. How many factors? 13 factors are responsible to produce one product. If any one of these factors are missing, or if any one factors are defaulted, then the coagulation process will not be accomplished. Understood? So all these 13 factors formation has to take place so that the end product, that is an inactive fibrinogen, is converted to fibrin. When a fibrinogen, okay, I will wait. When a fibrinogen is converted to fibrin, okay, so that will help in formation of this coagulant, okay, or coagulation process. To produce this fibrin, you need all these 13 what what do you call factors. Okay, all these 13 factors are essential. Did you understand, people? Okay, so we have to study the process of coagulation, but we have 3 minutes. Let's see what we can do. Coagulation of blood, as I said, it takes 13 factors to accomplish, and we can divide this coagulation of blood into two important categories, okay, or pathways. So that is called as intrinsic pathway and extrinsic pathway. Okay, see what is happening in intrinsic pathway and extrinsic pathway is, see, the intrinsic pathway happens when there is a damage to the wall of a vessel, whereas when I talk about extrinsic pathway, whenever there is extravascular cells, like because the body will be able to detect that there is an injury because of some antigens or because of some foreign substances present during the injury site, near the injury site, this extrinsic pathway will be activated. In very simple terms, we will try to conclude it in 1 minute, okay, and we will revise it in the next class also. Okay, so let's see. So the factor 12 will be converted to active factor 12, and active factor 12 will help you converting factor 11 to factor 12. Factor 11 to active factor 11, and active factor 11 will convert factor 9 into active factor 9, and active factor 9 will convert, okay, factor 8 into factor 8 into active factor 8, and finally the factor 8 formation will help in converting factor 10 into factor 10A. So now I know it's quite complex. So first one, what happens is you have your Hageman factor, which is produced by the liver. So this Hageman factor is called as your factor 12. So this factor 12 basically is in the inactive state because of the injury. Now this inactive factor 12 will become active factor 12. So this active factor 12 will convert your factor 11. So this factor 11 is called as anti-hemolytic factor C. So inactive anti-hemolytic factor C will be converted to active hemolytic factor C. Very, very important factor. Okay. Now, once you have factor 11, now is the time for factor 9. Now factor 9, factor 8, see, factor 11, factor 9, and factor 8, all of them are anti-hemolytic factors. Now, once we have factor 11, we have factor 9. The inactive anti-hemolytic factor B is converted to active anti-hemolytic factor B. Next, it will lead to the formation of active anti-hemolytic factor A. Did you understand? So first we had Hageman factor, it became active Hageman factor. Then we have anti-hemolytic factor C, it will become active anti-hemolytic factor C, that is factor 11. Next we have factor 9. That is anti-hemolytic factor B will become active anti-hemolytic factor B. Then we have anti-hemolytic factor A. It will become active anti-hemolytic factor A. And then how does it become anti-hemolytic factor A? This will form a complex. 9 and 8 will form a complex to produce factor 10. So it will not produce factor 10. It will convert inactive factor 10 to active factor 10. What is factor 10 called as? It is called as common factor, or it is also called as Stewart power factor. Very, very important factor. So why I tell this is a very important factor, because both intrinsic and extrinsic pathways, both of them will give rise to your factor 10. All right, students, let me not rush this coagulation process. Our time is up. Okay, we'll end this session now and we'll continue with coagulation in the next class. Thank you very much, students. If you have any doubts, you can drop me the message or any doubts in the chat box. We can look up to it in the next class.